Check it out now, y'all.
NanoHub U, online instruction.
All right, welcome back. Let's continue on the genome sequencing issue. I told you about how the genome sequencer works in lecture 28, and in lecture 29, we just discussed a set of issues like PCR, emulsion PCR amplification, which allows for better selectivity. Now, there are a whole sets of other issues that one has to think about also has to do with selectivity. And let me continue on those. So the next problem I want to explain to you is something called a homopolymer sequencing problem. This is again a selectivity problem. And you will see that most of the things that we are talking about now has to do with selectivity. Genome sequencing has solved the settling time and the sensitivity issue. No problem with those. But we are talking about now the selectivity one, which is really crucial. So let's talk about the homopolymer sequencing. Remember that within each well that we discussed a few minutes ago, the bead contains the DNA sequence. Let's assume that there is this base pair sequence that we are trying to read, and we are reading it one by flowing different bases in and gradually one by one building things up. So the C got conjugated with G, G got conjugated with C, and so on and so forth. And we have come up to this point where we have just built A to T. So now let's say I bring in C. I bring in C in solution, which is good. It will conjugate with G, and it will release one proton in the conjugation process, and the proton will diffuse down and will make a change in the signal, which is what I want, no problem. But you see, my trouble is that as soon as that conjugation is done, the C's are all floating around, and it will see that there is another G it can react. I really don't want to react it yet because I want to do it one by one. But you see, there are two G's sitting next to each other. As soon as the first reaction is complete, it takes a little bit of time. After that, it will immediately start reacting with and the G following it. And as a result, what's going to happen that whereas you had when things were going one by one, you have a certain level of signal, let's say. When you have the two G's sitting next to each other, then the signal will be double. Two C's sitting next to each other, the signal will be double. So you say, what's wrong? I mean, you know, if I have two G's, it's even better that I can read two letters at the same time. There should not be anything wrong. It turns out that it is really not that simple, as I will explain in the next slide, that there is a selectivity problem associated with it. Assume that I want to read this particular sequence. This is similar to the problem as before, ATTT, 4 T's, ACG. Now you can immediately see why it's called homopolymer, because you see I have a bunch of T's next to each other. These are the DNA bases all connected together. It's a polymer with the same basis, conjugative basis. All right. If I didn't know anything, then if I bring in A molecules, A base pair, then I would have expected a pulse of width 4, because first the reaction would have taken place. It would have given out a bunch of proton. Next one, a bunch of proton. Third one, a bunch of proton. Fourth one, a bunch of proton. If I added all the bunches together, it will be 4 times as high. So I should have gotten a signal which has a height of 4. But you see, that's not what's going to happen. The first pulse will look like that. Total composite pulse is actually going to look like the red one, the red one. Why is that? And by the way, And the red one has a height of 3. And the height of 3 means I will now be telling that I have actually T3s, whereas in reality I have 4. So I just made a mistake. Why is it 3? Why isn't it not 4? It is because that when you had the first signal, it came and it went. The second signal started with a slight delay, so that came in and that went. The third one is further delayed. So as you add up more and more of these signals with slightly constant amount of delays or fluctuating amount of delays, what you will see that gradually it will not add up at a single point. It will be shifted, and as you keep adding more and more, the height will sort of saturate, right? And then in that case, you'll be making a mistake. And this mistake is called an homopolymer mistake. And you can see the longer the sequence is. By 4, you are reading 3. If you had 5 or 6 or 7, you may read 4 or 3 smaller number. And therefore, this would be a big mistake because you want to read everything properly without sort of missing any base pair. It turns out that it depends if you don't rely on the peak, because if you are relying on the peak itself, then you can actually make a big error. Instead, if you integrate the area under the curve and rely on the area under the curve, the chances of making mistake is somewhat lower because the area under the curve is proportional to the signals more so than the peak, only peak itself. Of course, the longer time you allow, more accurate will be your estimate about how many T's did you have, how long was the homopolymer. But of course, you cannot make it too long because then you will be reading forever. In each well, you have to read many sequences down the road, so you cannot wait forever also. So if you wait, for example, on the right-hand side, if you wait 3 seconds, which is the red line, it turns out by the time you have 5 homopolymer in a sequence, you have tremendous error on 10% level of error in reading, which is a very bad selectivity and very bad, it will have very detrimental consequence. On the other hand, if you increase the time from 3 to 4 seconds, you can immediately see the error is considerably reduced because the area under the curve would be significantly better. And it turns out experimentally, you're somewhere in between. The point is that if you allow 3 to 4 seconds per read, then actually you can address this homopolymer problem quite well. The next problem that we have to deal with, it has to do with contamination issue. Remember I said the red well is loaded and the surrounding green wells, they should be empty, right? They should not have any signal. But you see, that's not how it's going to happen. If you look from the top view, let's say these are arranged in hexagons, what's going to happen is that the signal that has been generated, the proton that has been generated in the red well in the middle, going to come out after a while and going to contaminate its neighbors, which are reference wells. And the reference wells should not have any protons to begin with, because so that it's uncontaminated, can act as a reference. So the question immediately is that from these beads, as the molecules escape, what is the probability that it's going to come and contaminate the neighboring reference well? We can again do the calculation relatively easily. These are simple calculations, but I want to show you the final result. It turns out that the fraction that actually escapes forever through the global in the fluid versus the fraction that comes in and goes into the neighboring well is actually very small. And it turns out that the neighbor, essentially the amount of change in the voltage in the neighbor is minuscule. And so therefore, it turns out that even if you make things very small, very compact, the contamination is no more than 3 to 4%. And as a result, you will not be making a big mistake. But one must sort of make sure that you account for this couple of percent error so that this sort of the reference electrode signal can be appropriately subtracted. All right. Now, one thing I didn't emphasize when we are talking about pH sensing, that pH sensors or ISFET, right, ion sensitive field effect transistors, had this salt on the oxide surface, and there were these exposed bonds, the OH bonds, remember, and the pH sensing involved essentially exchange of OH and hydrogen atoms on the surface itself. Now, one trouble with salt is that salt diffuses through the oxide very well and or very fast, and it turns out that if you don't do something special, your transistors will be dead in an hour or so. So if the genome sequencing requires more than that time, you might think that you'll be dead because this signal can no longer be trusted. In fact, there are many experiments which shows that if you don't do not anything, just let's say you put sodium chloride salt on top of a transistor, because of the salt diffusing, your voltage keeps changing. So therefore, the salt essentially contaminates the signal. Again, it's a selectivity problem. And the issue is that, let's say your pH is changing from 8 to 7 to 6 and so on and so forth. Next time you go to pH 8, these two values may not be the same. Why not? Because by that time the salt has gotten in and it has changed the threshold voltage, and therefore the same pH signal now produces different responses. Something that's not really very good, right? So how does this problem is solved in the genome sequencing, which requires many hours to complete the operation? The way it works is that these wells, which are shown here on the top, unloaded, all of them are unloaded, the well is actually separated from the transistor by an interconnect, metal interconnect, and this is called an extended gate approach, where the gate has been separated from the oxide itself, and as a result, and the pH sensitive layer, the OA, silicon dioxide bonds and other things, these are all here connected through a metal to the transistor, and this extended gate isolates the salt, this is salty, from the transistor, which is very sensitive to salt degradation. There's no discussion between these two, and therefore, except the electrical connection is there, but the salt cannot come through. Therefore, this allows the reliability of the transistor underneath. A classic problem of ISFET thereby is solved. Finally, let me tell you a little bit about the scaling challenges, that if you wanted to make this thing much, much smaller, right? Every technology, remember, starting from 90 nanometer, there's a 22 nanometer technologies, people are talking about 10 nanometer technologies. So therefore, the transistors are getting smaller. Does it mean the genome sequencing machines can get more and more and more powerful? It turns out one has to be a little bit careful, because yes, I can go from the 65 nanometer technology to 22 nanometer technology. So I already have 165 million wells in here. The thing is, going smaller can be detrimental, and we have to be a little bit careful. It turns out that there are two issues, whether to make the wells smaller and whether to make the transistors smaller. You see, if you make the transistor smaller, then you have to have a special considerations. Let's say you have some reference size and you want to reduce the size by a factor of 4. Now, if you look at lecture 28, you will see the peak signal will not change. As you make the well smaller, you have fewer number of protons, on the surface, because the surface area, the number of protons, number of DNAs that you have is proportional to the surface area. Yeah, so therefore 4 pi r squared, the number of DNA per bead becomes smaller. That's true. But at the same time, the well is smaller. So therefore, the pH rise is about the same. And so therefore, the signal, which is the red line, will remain the same. On the other hand, if you look at the transistor, if you make the transistor smaller and smaller, the noise will keep rising. I'll have an extra lecture on noise, so I'll discuss it. But for the time being, only thing that you need to remember that the noise increases inversely with the cross-sectional area of the transistor. So the noise goes up. So the signal to noise ratio will degrade dramatically if you scale both the transistors and as well as the well. So which is a bad thing. Instead, if you keep the transistor size more or less the same, but decrease the well size so that you can pack more of them, that's fine, because in that case, the noise will remain invariant. So the point is that if you try to scale the transistor, it may not be scalable unless you somehow reduce the noise. But you can keep scaling the wells if you keep the transistors more or less of the same dimension. All right. So I told you about how to read all the pieces. So how you have a book. We have sort of torn apart the book in 100 million copies, give 100 million people to read them. Now the real problem is we have to put them back in together. So that chapter 1 is before chapter 2, chapter 2 is before chapter 3. This sequence, how do you put them together? You have read them in random order. How do you put them together? I will conclude with one slide discussing that effect, discussing that approach. Assume I have a sequence, original sequence, given here in the vertical line and shown here in this specific DNA. Now let's say after PCR amplification, I have made many copies and I have divided the copies in small chunks, small pieces. You see, I can either do the division after PCR amplification or before PCR amplification. That's fine. So this is one way of dividing. Remember, there will be 40 million of these. So this is, I will call experiment 1. So this is one set of experiment. It will take about a couple of hours to finish this experiment. Next, you do the same, repeat the same experiment, but cut it randomly. And since it's random cut, there will be 40 million pieces, but they will not be the same pieces. So this is experiment 2. And then there's an experiment 3, which is again 40 million pieces, but the pieces are not exactly the same as before. Once you have these 3 sets of experiment, right? This is sort of redundancy experiment, then you can put the original thing back together. This is how it works. Assume that in the first experiment, you got these fragments, AAATCG. Look at this sequence, AAATCG. So that's the first sequence. And then you got TCGA, TCGA, which is the next segment, let's say. So from experiment 1, you got these, and of course you will get many more. I'm not showing those. You'll have 40 million of these raids. Now, in the next experiment, in experiment 2, you'll again get another random sequence. Let's say in the experiment 2, it starts with CGTCGA, that's one. And then you have another sequence from the experiment 2, and so on and so forth. So you have a couple of experiments. Now you see, the way you can put them together is that this segment of the blue has overlap with the segment in the red. So therefore, the coupling pieces which will piece them together tell us which is after what. will come from the additional experiments, and you can see the CG has an overlap. So by looking at this first three, you know that this sequence has to be with this, because otherwise this has the match with the next one, and correspondingly this has the match with the next one. And by putting them together in the computer, by searching back and forth, you can put together the entire sequence. So my bottom line is that in order to put things back, you need multiple experiments, 40 million here, 40 million here, 40 million here, in order for this reassembly to take place. So let me conclude then. I wanted to emphasize that this pH-based genome sequencing are very new, relatively new, just a couple of years, maybe 2-3 years old now. But it allows, because it's electrical, allows the nanotechnology, is sort of based on the nanotechnology, relies on the nanotechnology to get high density. So therefore, it could be done its sequencing could be done at considerably lower cost. Now, like MOSFETs, when you make things small, there are many surprises. Now, one of the surprises which I alluded to towards the end is that you should not be scaling the transistor size too much, because the noise increase essentially compromises the signal to noise ratio. Right? Now, one of the concerns that often was that crosstalk between the loaded well and the reference well. It turns out that the crosstalk among wells, if you do the calculations, is really insignificant. When the protons come out of individual wells, they essentially get diffused in the bulk solution, do not really go, a large fraction of it doesn't go and contaminate the neighboring well. Right? And I tried to explain that why bead loading efficiency is so bad. If you want to have 4 unloaded wells next to every loaded well, then you can see the bead loading efficiency cannot be more than 20%. If you are somewhat less restrictive, then it can be up to 36%. The point is, loading more than 1/3 would be very difficult. And so therefore, although you may have 165 million wells, only 1/3 of them will actually be useful, which is fine maybe, because you need 40 million. So you may be more than fine. But the thing is, large fractions of the wells will do nothing and will be loaded with empty beads. And the point is that although the sensitivity is quite good, but one of the limitations of the approach that I said, that it needed PCR amplification, right? I had to amplify the signal. It would be much better if I didn't need to amplify the signal, if I could work with single DNA and get rid of the emulsion PCR. It turns out that for that, we may need a new generation of sensors which are far more sensitive. And remember, beating the staining limit and sensitivity limit in there, we talked about super NAS sensors. And maybe in the future, one might actually use such sensors to simplify the scheme even further. So these 3 lectures on genome sequencers, you should note that how it has brought together all the concepts that we have learned from lecture 1 to lecture 27. All these issues about settling time, sensitivity, selectivity, the lecture that I just discussed. It is very important that these pieces, you realize how these pieces come together, and then see the beauty of the technology, nanobiosensing technology, in its ability to bring everything together in a comprehensive whole. In the next lecture, I will wrap up the course. The next lecture, I'll simply summarize what we have discussed and what are the things that we should be looking forward to. Until next time, take care.